Hi, thanks for checking out my video. I know the other one that you probably have seen does not have audio, but so I decided to make this other one where we are gonna try and test out this application I built. So what does this do? So this application is supposed to help you sort of look for the most relevant information just using text queries. Like, just ask a question here and then it's supposed to answer questions based on these documents here. And just as an example, okay, just as an introduction, what's inside here is these are my lab reports that I wrote while I was working in a microbiology lab and a general research lab. So these are mostly relevant to someone who works in the medical lab. And let's ask a question to do with bacteriology. And the most common thing you're going to do in bacteriology is you're going to do a gram stain. So you can ask my bot, how can I that's not a good one. What steps do I need to perform the gram staining procedure in each step? Please add an emoji okay, let's just try that. And then what's going to happen is there is a representation of for instance, okay, this text will be turned into a bunch of numbers, and in the background this whatever text is in this document is also going to be made into a representation, which is something called an embedding, which is just your text is converted into like numbers with special stuff, and this is going to be stored in a particular database called a vector store. And within that vector store, we are going to do something called similarity search. We are gonna take the text that we have converted into an embedding over here, and then what we are going to do is we are going to query that. We are going to find the similar. So when you ask a question like this, you're going to look for documents with this similar text, in particular gram staining procedure. Then it's going to be found using the similarity search, and then you're going to ask a large language model like the ones for OpenAI or other ones you might have heard of about the question. And what you're going to do is they're going to help you retrieve that information, and then this information obviously is ranked already, and all what's going to happen is like it's going to put it together in a coherent manner like this, and then you can now get an answer to a question. I hope that's a good enough explanation. But yeah, so in summary, I just say this tool helps you find information just using text, as you can see here. And we've gotten the steps of how to do the gram staining procedure with some cool emojis. And yeah, that's basically it. So if you have questions regarding, maybe you have thousands of lab protocols, like this tool can help you like find the relevant information really quick. Yeah, and cool thing is you can also download your chat history over here. But before we do that, let's go to the molecular biology lab. And what do we do in molecular biology? We try and amplify DNA. That means make copies of DNA. So maybe you've forgotten what are the requirements, or how to amplify your DNA. So you can ask, how do I perform polymerase chain reaction. So this technique, it helps you to figure out, it helps you like amplify your DNA. That means make more copies, because in most of molecular biology experiments you'll need more DNA to do maybe another step in your process. Maybe you want to do sequencing, maybe you want to identify a particular microbe. Like there's another procedure called real-time PCR. That's not a good example, but there is something called there's this diagnostic test that people do with TB, and there was a certain tool that people used to use for that. And as I try and remember what that is, let's just ask this question. So let's just search that. Then in the meantime, let me see if I can remember what I used to do with the yeah, let me just try and remember. There was a test people used to do that helps you identify Mycobacterium tuberculosis, which is a bacteria. And there was this real-time identification thing that uses this technique. So I can't really remember what that is right now, but maybe in the description I'm going to add it. But apart from amplifying DNA, it can also identify the microbe, which is some sort of a real-time polymerase chain reaction. And yeah, that's basically it. Like it had a really cool name. I just forget what that name is. But let's just review what we have here. PCR buffer that's needed, magnesium chloride, DNTPs, primers, template DNA, Taq polymerase, and double distilled water. I just remember this is how I wrote my notes exactly within one of this, and it's kind of like a blast from the past. And also with this technique for grabbing relevant information from these documents, you can think of it like a search engine. Like what happens here is that if there's any mistakes in this document, like it's probably going to propagate also here. So I'd advise you to probably write your protocols really well. Otherwise, this is the application. I hope you liked it. And if you have any questions, you can just either comment on the video, or maybe if your lab is interested in it, you can just let me know and I can make something for you. And yeah, and just to confirm that this actually works, let's just click on that, and there's my chat history. Let me just call it chat history 2. Thanks for watching. Bye.